The epidemiological observation that populations eating fatty fish regularly show substantially lower rates of dementia than comparable populations without this dietary pattern has been documented across multiple large cohort studies, with some analyses finding 30 to 40% lower dementia incidence in high-consumption groups. The explanations for this association have historically centered on omega-3 fatty acids, particularly EPA and DHA, and their roles in neuroinflammation reduction and neuronal membrane fluidity. More recent research has added vitamin D to this conversation in a way that appears mechanistically complementary rather than competitive, and the combination of vitamin D and DHA in the same food matrix, as found in wild salmon, mackerel, sardines, and anchovies, may explain why whole fatty fish consumption outperforms omega-3 supplementation alone in some cognitive outcome studies. Vitamin D, through VDR binding in neurons, upregulates the autophagy genes that enable clearance of the protein aggregates, amyloid beta and tau, that accumulate in Alzheimer's disease and frontotemporal dementia. This neuronal autophagy mechanism is distinct from the immune cell pathogen-clearing autophagy discussed in other contexts, though the same VDR2 autophagy gene pathway underlies both. The relevance of this mechanism to cognitive aging is that amyloid beta and tau are continuously produced in aging neurons and continuously cleared through autophagy under healthy conditions. When autophagy efficiency declines, as it does with aging, with vitamin D deficiency, and with mitochondrial dysfunction, these proteins accumulate and eventually reach the concentrations associated with pathological aggregation. Maintaining adequate vitamin D to support neuronal autophagy gene expression represents one of the more mechanistically coherent nutritional strategies for preserving the protein quality control that prevents this accumulation. DHA's contribution to this system is structural rather than transcriptional. This omega-3 fatty acid is incorporated into the phospholipid bilayers of neuronal membranes, including the membranes of autophagosomal and lysosomal structures. Higher DHA content in these membranes appears to enhance autophagosome-lysosome fusion, the critical step where the autophagosome delivers its cargo for degradation. Neurons in DHA-deficient states show impaired autophagic flux even when upstream autophagy signaling is intact, because the membrane composition prevents efficient autophagosome completion and fusion. This helps explain why DHA supplementation alone produces modest cognitive protection, while dietary fatty fish, providing both DHA and vitamin D together, shows more consistent associations with cognitive preservation. The practical protocol involves 3 to 4 servings of fatty fish weekly. Wild salmon provides approximately 988 IU of vitamin D and 2.2 grams of omega-3 per 3.5-ounce serving, making it the most nutrient-dense single option. Sardines are the most economical at 193 IU vitamin D and 1.5 grams omega-3, with the added advantage of being a small, short-lived species with negligible mercury accumulation. Mackerel provides 360 IU and 2.6 grams omega-3. Anchovies are concentrated but typically consumed in small quantities as a flavoring. Mercury concerns are real but manageable through species selection. Small fatty fish accumulate dramatically less mercury than large predatory species. For plant-based eaters, the combination of algae-based DHA, the original source of DHA in the marine food chain, with vitamin D3 supplements targeting 40 to 60 nanograms per milliliter serum levels, provides the most direct replication of the two-mechanism neuronal autophagy support that fatty fish delivers.